What is up, Brad fans? Welcome back, and thanks for joining the show once again. Uh, Very pleased to bring you this conversation. I know you'll enjoy it. I was joined this episode by Stephen Doyle, who is a molecular and computational biologist working with parasites, something I'm very familiar with, a topic I quite enjoy. So, Steve is a group leader at the Sanger Institute in the UK, Sanger Institute being one of the biggest labs for genomics, gene sequencing, all of this kind of stuff. And that's one of the reasons I reached out to Steve. I followed him for a while on Twitter. He's very immersed in the world of the latest in terms of genome sequencing, gene sequencing, and then he applies that to parasites, which we will talk about, looking for resistance genes, how parasites can evade the drugs that we throw at them. But Steve is also just well versed in all of the sequencing technology, and so he was able to kind of help us break down some of the things that we've been hearing about in terms of variants with the coronavirus, molecular epidemiology, how you use genome sequencing to sort of track an outbreak, which you may have heard about with this whole coronavirus situation. So yeah, very knowledgeable in that stuff. Really enjoyed speaking with him, and I think you will hopefully learn a little bit about some of these terms that you're hearing thrown about maybe in the news. We also talked about the importance of open science, and Steve had some comments about preprint, um, publishing preprint papers, which, if you remember a couple episodes back, I spoke about with Dr. Martin Nielsen, also a parasite guy. Um, and Steve has kind of a different take on it, and he laid out why, especially for his field of genomics, that preprint works really well, and that it's advantageous. Now, I don't think Martin would disagree with any of that, but they did have differing opinions on it. So it was Steve had reached out on Twitter to kind of start a discussion on some of those things that Martin and I had talked about, and we continued that discussion here. So he's got a really interesting view on some of the science communication stuff, how to get that out there, how to get science out to people, make it more accessible, not just to the public per se, but to people in low and middle income countries. That was something he brought up as an avenue of science communication that I had never really thought about. But that is another um, lane of science communication that you could get into as an academic. And he explained some of the projects he does, trying to share knowledge and share resources and stuff with places that might not have access to some of the financial resources or laboratory resources that other places have. So really great to have Steve on the show. You can follow him on Twitter. He's a great follow on Twitter, adding some, like I said, some explanations and some comments on all of this genomic stuff. If you have any interest in genomics, bioinformatics, gene sequencing, that kind of stuff, he's a great guy to follow on Twitter at Stephen __ Doyle. So that's Stephen with a PH, STEPHEN __ Doyle, DOYLE, Stephen Doyle. And of course, follow us at 2brad4u on Twitter and Instagram. You can hit me up at bvamparadon at Twitter and Instagram. Send the show an email to bradforyou@gmail.com, or leave us a voice message. We will listen to them and read and play them on the show. Speakpipe.com/2bradforyou is where you can leave that voice message. All of this is available on our website, 2bradforyou.wordpress.com. Really, really hope that you reach out and become part of the show. That would be, nothing would make me happier. Yeah, and then just subscribe and comment, rate us, all that kind of stuff really, really helps. So that's it for me for now. Please enjoy my conversation with Stephen Doyle. Steve, thank you so much for being here. Thanks for joining the show this Monday afternoon, taking some time out of your day. It's really nice to meet you. I followed you on Twitter for a while.
It's my pleasure. Thank you for inviting me. I mean, it's really excited to to chat to. I have followed you for a long time, both when you were doing science and since leaving science, and so it's great to be a part of this side of
Yeah.
things.
Yeah, very cool. Yeah, I appreciate that. I think, the listeners will probably, your accent probably gives you away. At least it gives you away to me, but you're originally from Australia, correct?
I am, yeah, originally from Melbourne, born and bred. Yep.
Yeah, and now in the UK?
Yeah, so we moved to the UK in 2015, and so yeah, just over 5 years now.
It's because, and we'll get into this, you're at the Sanger Institute. That's in Cambridge, correct?
Just south of Cambridge, about 20 minutes
Okay.
south of Cambridge, out in the green fields in the middle of nowhere.
Yeah, right. I've been to Cambridge a couple times, and it's always, people always say to me that it's like this sort of window in the UK where you actually get sunshine
Yeah,
and whatnot a lot of times during the year and stuff. So it's got this like weird weather pattern where it tends to be a lot sunnier than most places in the UK. So
I was really prepared moving from Australia. Melbourne hasn't got the best weather all the time. It gets hotter in the summer and not as cold in the winter, but I was prepared for it to be wet and miserable. And actually, this area is one of the driest in the UK, and so
yeah,
we don't get a huge amount of rain, really.
it's surprising.
Yeah, it's
I
nice.
got one more thing on the sort of Australian moving to UK thing, because I don't know if you've been to Canada. I'm originally from Canada. There's a lot of, I feel, synergy between Australia and Canada. Even though we're on opposite sides of the world,
Yeah.
we have big countries, sparsely populated, weather and the elements. For you guys, it tends to be hot. Us, it's cold. Play a big part of life. And then obviously the former British colony thing, so I don't know if you've noticed this, but for me, my age group of people, there was always people going to like live in Australia for a year. And where I'm from in Canada is where all the ski hills are, the mountains and stuff are, and it was
Yep.
just filled with Australian kids coming to work and ski in the mountains and stuff. So
Yep.
I've always felt there's this synergy, this like we get each other even though we're so far apart. But I have to say, I feel like you guys tend a little bit more towards British culture, and we're kind of a little, we're so influenced by America.
Yep.
Was it that big of a stretch for you to go to the UK, or do you see
No,
those similarities? Am I way off?
no, I think it wasn't a big stretch. It wasn't a culture shock that you'd get moving to certainly other places in Europe, for example. I mean, we didn't have to deal with a language change or anything like that. I think Australia and British humour is quite similar, and so that we fit right in a lot of ways.
Yeah.
So yeah, no, it wasn't such a big difference. I think a lot of people were shocked that we moved over here, especially to the sort of small country town that we live in, and they would notice the Australian accent very quickly and then say, "Why did you move here?" Shock horror, because I think everyone thinks Australia is sunny all the time and we all live on the beach. And well, a lot of people, everyone lives the majority of the population lives within a relatively short distance of the coast. But yeah, being from down south, being in Melbourne, it's really variable weather, and it's actually not too different to where we are here in the UK. So
Yeah. Yeah, it's funny. I get the same thing here in Germany when we say we're from Canada. Like, I'm always shocked because the German people are pretty blunt, and they'll just say, "What are you doing here?
yeah, exactly.
Canada's great. Why did
Yeah.
you move here?"
Yeah.
And then you got to be like, "Well, your country's not bad either. There's some
Yeah.
opportunities here that we don't get other places, and Europe's pretty cool and stuff like that." But they're just like, "Why are you here?
Yeah.
Why would you leave Canada?" It's
Yeah.
got that. And I think Australia has that reputation as well, of like these sort of paradise kind of places where everybody gets along and things are good. And
Yeah.
obviously, every country has its own plus and minus, pros and cons. But
Yeah.
anyway, we got in touch on Twitter because we started a Twitter conversation based on an episode I did previously with Martin Nielsen, who is also a parasitologist. You work with parasites. I used to work with parasites. We're all in this field together. And you had mentioned some of his comments about preprint. So for to refresh people who maybe listened to it a while ago or hadn't listened to it, Martin and I were talking about science communication sort of broadly, generally, and he mentioned this sort of, I guess it's a rather new phenomenon, we'll say, of preprint servers. So rather than it's basically a place where scientists can publish their work before it goes through the sort of rigorous peer review process. And the peer review process is just getting other people in the field to critique the work, suggest changes, that kind of thing. And then, the editors of the journals decide whether, okay, this has been, sufficiently reviewed and now it's published. So we kind of like, that's sort of the gold standard of like, now this is out there in the scientific community. Whereas preprint, it now offers people a chance to publish online before going through that process, which has its pros and cons. So Martin wasn't a big fan of it. He thought, you can go back and listen to that episode, people, if you want to get his take on that. But there is pros and cons to this process. So Martin wasn't a huge fan. Kind of felt like we really need that peer review process. And if people don't understand that these papers that are out there on the internet on these preprint servers, if they don't understand that it hasn't gone through that sort of rigorous sort of checking process, you have to treat that information differently. And then you jumped in to talk about some of the pros that you thought were there. Some of them may be a little unique to your field, which is genomics broadly,
Yeah.
in terms of getting information out that everyone can share. So maybe you want to jump in here and just kind of lay out your kind of case or what you find attractive about the preprint process.
Sure. So I guess the best place to start is why I think open science is so important to science in general. It's important for researchers. It's important for the public. I think that we need to be as transparent as possible, and the way to do that is to try and be as open as possible. Because a lot of science is competitive. It is done behind closed doors. It's not always reported in the way that it should. And part of that competition is because we're going for grants or going to try and publish papers in journals that we believe are reputable. And we, as scientists, we pay money, or the university pay money to these big journals to have the privilege of publishing our own work and having it in a glossy journal. And so there's all this competition, there's all this money involved, and a lot of that really ultimately restricts science being done. The scientific publishing model is not equitable because there's money involved, and not everyone can publish in the highest impact journal, regardless of whether their research is perfectly suitable for publishing in that journal. So there's one problem with the current journal system, and the second one is that not everyone can access research in those journals because there's a subscription model. Some journals require you to pay money to either access an individual article, or a university library or institute will pay quite a substantial amount of money to the journal to allow their researchers to have access to that. And so you can now imagine that there's actually a large proportion of researchers in the world, particularly in low and middle income countries, that just do not have that access. And so preprinting is one way of allowing scientific research to be put into a public domain that doesn't cost any money, and it's open access. It allows anybody to access that information. And this has been around for a number of years. It's not necessarily recent, but it's perhaps more recent, if you like, in medicine and areas like genomics that I work in. Certainly, I don't even want to put a date on it, but I've been publishing in. I've been putting our papers in one preprint server, bioRxiv, for at least 5 or 6 years, maybe slightly longer. I think 2015 or 2014 was the first time we used that. But in fields such as physics and mathematics, these preprint servers have been going much longer and are much more accepted in their communities. And so while it feels new and strange to some, I think that there is a real shift in perception about their use. And the discussion you were having on the previous podcast was really around one negative aspect, in that because it's open, and it's because basically anyone, I'm going to say that with virtual inverted commas, anyone can publish it, that there's perhaps some problematic research in preprint servers. And I think that's true to a degree. But at the same time, there's problematic research in, Nature or Science or Cell, really big journals that people pay thousands of dollars or pounds just to publish it. I mean, now I think it costs close to €9,500 to publish in Nature. That's just for one paper to go through the publication process. It's completely inequitable. So so there's problems in peer review. It's I have published quite a few papers now. I've reviewed a lot of papers as a peer reviewer, and I'm also an associate editor for Parasites and Vectors, one of the journals that publishes a lot of the research that I guess I read and I'm interested in. It's not a high tier journal, but I've published a few of my papers in Parasites and Vectors. And so I've seen this from multiple angles, all of the angles that you can think of the process. And and I've got enough experience now to know that peer review is not perfect. There are ways, not ways that you can game the system, if you like, but there are certainly ways that you can tell that it's gone through peer review very easily when maybe it shouldn't have, and there's other times where papers will get hard done by because a particular reviewer has a problem with it for a good or not reason. So in terms of the good things, I think that are missed, but that perhaps people don't realize about peer reviews, preprints, especially from someone coming from an early career research perspective. And I've only just started my own lab at Sanger, and I've been postdocing for a few years. And I've gone through this process where I've been in short-term contracts or short to medium-term contracts. I've moved from Australia to the UK. I've set up new research lines of inquiry. All of these things take time. And one of the problems about publishing all of that work is that also takes time. People perhaps don't realize that to publish a peer-reviewed article, it takes a certain amount of time to do the research. It might be a year or two. It takes time to write the paper. It takes time to get all your co-authors to sign off on it. And then once you press submit on a journal, or submit to a journal, then it might take weeks before the editor even responds properly. It might get desk rejected, which means that the editor just says, no, it's not a good fit for our journal. But if it does go out to peer review, peer reviewers usually have 2 to 4 weeks, maybe, sometimes shorter, but often 2 to 4 weeks. So you're waiting a couple of months. They're going to get some peer reviews back. It might take a few months to make the corrections. You resubmit it, takes a few more months. It could be a 6 months to 12 months process between pressing submit when you think your research is ready for the world to see and it being published in a glossy journal. Now, what a preprint does is it basically allows you to make that visible from the get-go. Now, in my experience, I've submitted to a journal, and on the same day, or essentially the same day, I've submitted to the preprint server. So what I believe as my research is good enough to submit to a journal, then it's good enough to submit to a preprint. And what that does as an early career researcher is it allows you to be visible. If you're applying for grants or fellowships or your next job and you have research that's not published, it's not visible. No one can see that at all. You don't really have any evidence that you've done any of that. It's very difficult to make a strong case to say that you're the best at this when you don't have any evidence that you're the best at this. Whereas what a preprint does is it means that it's open, that a reviewer of your grant or your fellowship or your next job can click on the link, see what you've done, read it in the way that they would read a journal article, a published journal article, and can judge you on your work. And I think that is extremely powerful because, as I said, postdocs, so people who have done their PhD and basically in training to try and become an independent group leader, usually do one or a few postdocs. And as I said, they're 2, maybe 3-year positions. It's very difficult, even in that 3-year time, to turn around publications or multiple publications. But preprinting gives you that evidence. You can write that. A lot of journals now will allow you to cite preprints. Grants will allow you to cite preprints. And so it's an excellent way for an early career researcher to show evidence of what they've done.
Mm-hmm. Yeah, you bring up a number of good points of like how it benefits sort of, you mentioned, makes it more sort of equitable for people to publish, get their work out there, get it seen, because there's the financial restrictions, which, I think a lot of people don't realize, like if you're coming from outside of academia, which, a number of people that listens to this show I know do, it's like if I want to publish my paper in a journal and get it out there so that I can say, hey, I did this work, it's approved by science, you know. Yeah, 9,000 euros you're talking, for the top ones, and it's I think it's like the shortest I've seen is like 2,000. But you also bring up these really good points for the early career people, because I think that's another thing that maybe it's starting to trickle out into other mainstream areas, this idea of how science is done and how really like postdocs and sort of grad students are like this this labor force that is doing so much of these like experiments, and they're all like fighting for these positions and grants, and jobs, that there's not a lot there. So it's like you mentioned before, very competitive. But like this process of like how long it takes, and what if you're, I've heard so many horror stories of grad students where they want to publish something, but like it takes forever to go through the peer review process, or the supervisor is dragging their feet, or all of these things. So there's a lot of benefit I see for, within the field. Continue,
In my experience, I mean, everything takes us twice as long as you think it will take.
Yeah.
In science, everything takes twice as long. And someone in a 3-year contract, you really need to start applying, or at least put your job searcher hat on within your last year of your postdoc. I mean, it's unrealistic to think that unless you're really connected, if you're lucky, you might come from the right lab that has the right connections. But I think for a lot of people, it does take a long time to go from that one position to secure the next. I recently secured an independent fellowship, which is allowing me to start my own group at Sanger. And I submitted the fellowship application in May, and I found out that I was successful the following April.
Yeah.
I mean, that's 10, 11 months. And I could have found
your
out
life around that.
Yeah, I mean, if I hadn't have got that, then I would be now back in Australia because I would have had to move home. I didn't have time between the end of my previous contract, which was September last year, to apply for another round of that funding. So you've got to have backup plans. And as I said, the key thing is that everything takes time. And what preprinting does is that it allows you to make your work visible when you think it's ready to be visible.
Mm-hmm.
I think the other there's I can talk about a bunch of different aspects of why
Yeah.
there's benefits here, but another thing is that it puts a timestamp on when when you're ready to show the world something. And again, in this really competitive, you know, sector, if you like, science
Mm-hmm.
is competitive to a degree. We're all chasing a limited pot of money. There are limited jobs, as you say. Preprinting can allow you to say, hey, this is when it's ready, and this is the date that I've staked my claim on this. And a good example, perhaps, is the CRISPR battle
Mm-hmm.
between University of Berkeley and the Broad Institute, in that there was some overlap in the time at which papers were submitted and papers were accepted. If two papers are submitted to different journals on the same thing at the same time, the time at which they appear in publication could be vastly different. And one might
Right.
even be earlier but come out later. And so the
Mm-hmm.
preprinting does allow you to sort of stake your claim a little bit. Now that could be used in the wrong way, in that you could
Yeah.
have an idea and say, this is a fantastic idea, I'm going to write a preprint and not show any evidence. And therefore you'd be staking your claim before you had anything to back it up with.
And in that sense, you could see it as like, because obviously the critique or whatever will be, well, it's still going to, it's people will still see it and judge it on the value of its work. But
That's it.
unfortunately, if you're the first one out there and you get it out there, maybe that is what people remember, rather than, oh yeah, but actually it turned out that the work wasn't really that good, and then we had to like change it or something like that. It's almost like when newspapers make corrections, right? Like everybody remembers the headline and nobody remembers the correction that comes 2 weeks later
Yeah.
in the next edition or something.
Yeah, that's very true. I mean, there is a big thing about being the first, being the biggest. That has the impact. Part of one of the problems with scientific publishing is that we're in some ways, I think force is the wrong word, but we're naturally encouraged to try and make our research as biggest and brightest and spun in such a way that we've done something amazing. I think my work is amazing, and it's very easy to be brought back down to earth when you go to a conference, and there's a lot less people interested in what you have to say than what you think you have to say. But if you read really big publications, sometimes there is a lot of spin that's gone into it to make it sound really, really big, and sometimes that's what is attractive to those journals, because they want to be publishing the biggest and the first and the best.
Mm-hmm.
And
In a lot of ways, it kind of, it kind
Mm.
of reminds me of, and maybe an analogy for people outside of the academic world. It kind of feels a lot like how people have conversations about media these days, about like just needing to grab attention, grab headlines,
Yeah.
grab, this. So you have to like, you get the flashy headline, you get this, and then you read the article and it's like, well, that's not really what happened.
That's
There's a little
exactly.
bit of spin going on. And it's unfortunately, yeah, science can operate in that way too. I think within the science community, there's a lot more sort of, maybe we'll say, natural checks and balances, rigors that maybe tampen that down. So it's not, I'm not trying to say science is clickbaity, but there's that similar sort of pressure to sort of be first, be big, because there is, like you said, they're chasing us a limited amount of money. Big research is going to get big research dollars, which is going to allow you to keep going. It's just sort of this, natural evolution of the cycle that's been created. So
Yeah.
I like what you're saying about preprint as a way to help the younger people sort of, get that timestamp, sort of show like, look at, it hasn't come out, it hasn't gone through the whole process yet, but I'm comfortable showing it to you so you can judge it, whether it's for a job application, grant application, whatever, or fellow scientists, right? Like it allows everybody to get eyes on it.
Yeah.
And so instead of like 3 peer reviewers, you can have hundreds.
100%.
Yeah, so that's all good. That's all great. That makes sense to me. And in the end, I think bad science will get found out, whether
Yeah.
it's through preprint or peer review. The question then is, you know, for the, let's say, the general public or something, because I was thinking about this and I was like, preprint, who does that benefit? It benefits the science community, like you've outlined. I think journalists, it benefits as well because they can take a look at it. Now, this is one of the things that Mark and I were talking about. You have to understand the caveats of this hasn't gone through the rigorous peer review process, so you need to sort of report that responsibly. I think people are sort of learning that. But then it's like there's still this bridge to the sort of, let's say, general public, right? They're still relying on someone to interpret that stuff. Because I don't think the general public is going to preprints, right? And like looking this stuff. I mean, maybe some people are now in the age of Corona, because there's just like everybody wants information on how to be safe and what's going on and all that stuff. But that's, I think, where to Martin's point, there's potentially a danger there with the bad science getting out and stuff. And I'm just wondering what your take then is on, because you did mention sort of at the beginning that there is a benefit to the public. So I would like to hear what your thoughts are on that, because
Sure.
to me, there's still a disconnect between, preprint and we'll say the general public.
Yeah. I mean, this might be an unpopular opinion, but it's my opinion, is that science, but so, like everyone's opinion, everyone has one. But my opinion is that science journal, peer-reviewed or pre-printed manuscripts are not meant for the general public. Right? Mm-hmm. And this is maybe a big statement, but in reality, a published journal article or a preprint is written for other scientists. Okay, mm-hmm. It's used, the language that's used, the technical detail that's used assumes some level of technical training. It's if it was written for the general public, they would be written very, very differently. Mm-hmm. And so that's, I think, an important thing to say, is that they're not written for anyone else. And if anyone else reads them, there's a good chance that there's going to be some degree of miscommunication. Mm-hmm. So, accessing, making this accessible to the general public is the responsibility of things like media journalists, in communicating that correctly. And as I said, I'm a big advocate of open and transparent science. Part of that open and transparency is saying when something is being peer reviewed, when it hasn't. I think that is getting easier to interpret in journal media journalist media type forums. It often comes with a caveat of not peer reviewed. Does the general public understand what that means? I'm not sure. I guess it's part of the challenge of this ongoing science communication fight that you're clearly fighting the good fight here. But it's very difficult. I think we need to make sure that we're not spinning our science too hard, so that when someone else reads it, they don't spin it even further and make it something that it's not. And that's sometimes where the problems lie. There's sort of this Chinese whispers of what something actually means, when in a scientific paper it may be actually quite defined. It might be a cure for diabetes in a cell line or in a mouse, or something that has a long way to go to being translatable into humans, but it's very important that all of the details are understood. I was talking to someone the other day about understanding journals, and I really don't think until you're probably somewhere in your PhD. Okay, so this is the level of learning that's involved of how to really read and understand a paper, a published journal article. It takes a lot. You sometimes, almost always, not all of the details are there. So you've got to kind of work out how to read this holy document that's, it's not always clear what's being done or why something's being done. Some are better than others. Some of the statistics are poor. Sometimes it's really just complicated and the author hasn't written it very well. But I really think that it takes a lot of training and reading a lot of papers to actually become quite competent at reading a paper. And even having written some papers is probably a good learning curve too.
Yeah. I mean, I personally, I kind of agree with your point that like journal articles, like scientific journal articles, primary source stuff isn't really meant to be, widely consumed. I mean, if you want to think about, the goal of that paper really is so that other scientists can read your work, know what you did, replicate it, and build on it, right? That's the goal of that document, right? Is to provide a recipe, a manual, so that someone in the same lab, a similar lab as you, with the similar materials, can follow that recipe and see what they come up with. Make the slight change, see what they come up with, build on it, that kind of thing. So for 100%, like if you just look at like what the goal of the document is, the goal of the document isn't to get the general public excited about this research, right? Like that's the next level of it. Now, I mean, I think you could probably, maybe you would agree, maybe you wouldn't, that there's still a lot of work that can be done in terms of like how jargony and how technical these manuscripts are. There could be some work there to help, even just some cross pollination between fields, right? Like so that you get that sort of someone from a distant but still kind of related field might be able to find something in your work that makes this great cross-disciplinary discovery. But yeah, I mean, I totally agree. It's not really meant for the public. So then, getting back to the preprint, it's great then for the science communicators, the people that want to take that stuff, whose job it is to take that stuff and bring it out. But it's just, there's always this challenge of, the understanding of the sort of nuts and bolts of it, right? I did the last podcast I just did, I was speaking with a science journalist, a fellow science journalist, and she said, it's important to remember that science isn't a thing, it's a process.
Yeah.
Right. And I'm wondering how much of that understanding gets out there to people. I think, if you're trying to, the pandemic has highlighted so many, I think, failures in communication, where, things change and you have to now we have to change course, or we have new information or something, and that really throws people for a loop, and politics doesn't help, all that kind of stuff. So do you have a stance on like science? You're starting your own lab. Do you have a stance on science communication as to like, we're sort of encouraging this as members of our lab? It's been another conversation that's happened on these podcasts is, academics don't get usually compensation or credit for doing that work. But where do you feel that, the role of the academic is in that sense? And, if you do engage in science communication, I mean, you're doing it right now, but what sorts of things have you found useful, or would you try to target, that kind of thing?
I broadly think it's really important. So I have a huge amount of respect for what you're doing, and other people that spend their time, often not paid, to try and get people in, get the layperson to understand what they're doing. I think it's always been a two-way conversation, or a good science communication is a two-way conversation, in that you can hopefully convey a little bit about your passion, about what you do and why you do it. But also it can be a great way of learning about how other people, perhaps not in your field, or perhaps your mum or your dad or grandma, how they might understand what you do when a lot of the times you live in a little silo working on something kind of complicated that, maybe lots of people don't understand. Hopefully we all have as scientists, we have a greater goal of what we want to achieve with our science. I think it's much easier to sell you and your work and, to get grants when you have that bigger vision about where you think your science fits in the world. Because ultimately we don't work in silos. We work as part of this process, and that if we're lucky that we'll develop something, a paper or an idea or a methodology that forms one little building block in this great big world that is science. And if hopefully, if you're lucky, you'll get a couple of those that stand the test of time. But we don't know though. Things are changing really rapidly all the time, and sometimes our experiments are really complex things that we try and reduce down into something really simple, and it could be the fact that we've just simply missed a key variable that for some reason we just didn't appreciate that was important at the time.
Mm-hmm.
So that can be really difficult. So I've been active in some ways with science communication. I wouldn't say that I've done a lot, not for a lack of want, but one thing that I'm really passionate about, and it feeds into my work in some ways, is interacting with people in low and middle income countries, particularly around the development of skills and training to do with genomics and bioinformatics. And since joining the Sanger in 2015, I think every year I've been on at least one or more than one course that we run. It's essentially run through the communicating science and engagement platform that we have at Sanger, to take bioinformatics training from Sanger and deliver that in different parts of the world to people that really just don't have the access to that sort of training materials. And these are all bioinformatic courses that I've been on, and so we're teaching a bit of computer coding, and there's also a bit of context about talking about parasites or other pathogens. And these are targeted at trainees, people that are either, for example, in their postgraduate degrees, masters or PhD students, or early career researchers who are actively working with these sorts of data sets, but perhaps don't have the skills or aren't in the environment that allows them to do these sorts of things really effectively. And I get a lot of satisfaction from this, from talking to people, training these people. You always walk away with everyone wanting to collaborate with you, which is
not a bad thing.
Which is not a bad thing. But I think that's one way we've done that. As I said, I've been on courses every year, and we're running a virtual course in May in Asia. We were meant to go to Thailand, and there was another one that we were meant to go to Vietnam to run. So these are week-long courses, but we've been to Uruguay, South Africa, Ghana. We've been to Asia before, range of different places.
That's an really great. I mean, it's a great initiative, but it's an angle of science communication that I, to be honest, never even really thought about. But that's another aspect of this broad term science communication. I always think of it as media, science journalism, podcasts, that kind of thing. But actually, there's a number of different audiences that need this information, and you've highlighted a number of times in this conversation already that low and middle income countries don't necessarily. They're kind of being shut out a little bit for factors that don't really. That don't involve the actual work, right? Like it's access to some of the materials, some of the knowledge, some of the publication instruments, that kind of thing. So that's a really great point, I think, realize that that's another angle of science communication. Maybe now we can jump then to the work that you actually do, because you kind of you threw out a couple terms there, genomics and bioinformatics, which
Yep.
I think people, that follow science somewhat will have heard those terms, maybe understand what they are, but I think we could do a little explanation on that, because I think there's probably another group of people that have just been introduced to genomics and bioinformatics because of the pandemic.
Yeah.
And they're hearing these terms, variants and sequencing and all of this stuff. And that's sort of your wheelhouse. Now, yes, you work with parasites, maybe not so much viruses. I don't know, maybe you have some experience with viruses, but I think broadly, the methods are the same.
Yep.
The principle behind it is the same. We're all based on DNA and RNA and these things. So maybe you could kind of give us the sort of broad strokes, like genomics as a field, what it is, what it can teach us about whatever organism we're looking at. And then, we can kind of put that in context of some of the coronavirus stuff. And then we can also, your specific work with parasites, because from, I pretty sure, I don't want to jump on your work too much, but parasite drug resistance is a big part of what you do. And I think that's an important topic as well. But
Yep.
let's start broad.
Okay. So for a really long time now, I've been fascinated by genetic variation. So this is, I completely agree. It's variants and genomics and sequencing is all over the news, and I get really excited to see it because you don't see these sorts of words often used in mainstream media. But I'm fascinated by how we're all different, how we're one species, but we all look very different, and a very common thread for all life is that we have DNA, and DNA is a little bit, one analogy is a little bit like an instruction manual for how to build an organism, and the variation in that DNA, there's very, very small changes that differ between different individuals within a species, create all this phenotypic variation. So phenotypic is different from genotypic. Genotypic is those changes in the DNA. Phenotypic is what we see. So in people, we have different color hair
color,
and
eye
eyes.
color.
Yep,
Yeah,
exactly. And so there's an amazing, an amazing diversity of genetic variation within a species. And that's what creates all this phenotypic variation. And obviously it's in the news at the moment because we're talking about variants in the SARS-CoV-2 genome that might be more or less transmissible, pathogenic, these sorts of things. No, I do not work on viruses at all. But you're right, a lot of the same principles apply, in that understanding what these variants do can really tell us a lot about how an organism changes over time, how they acquire these variants, and where do these variants lie in their genome. So their genome is all of the DNA that's within a cell or a tissue or an organism. So how do these variants contribute to this variability? And what does an organism do with it? And how does that spread, or how does that move through time and space? And that's kind of what I'm interested in. And I work on parasitic worms, so called helminths, and these infect humans and animals, also infects plants. And they have an amazing diversity of phenotypic variability. They infect all different types of hosts. They infect them in different ways. They are able to live in different tissues and have really complex life cycles that might involve intermediate hosts, where to allow transmission from one human to the next, for example, it might have to be transmitted by a black fly or a mosquito. And they've got to be able to adapt and fend off all of these defense systems between hosts, and a lot of this is through evolution, and evolution acts on this variation to enable filling of a niche, if you like, that other organisms don't fill. So yeah, understanding this variation is quite fascinating, and parasitic worms are a particular variable. They're really highly variable.
Mm-hmm. Mm-hmm. So, like I think people understand that we now have this ability to sort of get the A's, the T's, the G's, the C's from a cell, from an organism.
Yeah.
So get those letters that we see that are associated with the DNA code.
Yeah.
But then, so that's, one aspect of it is the technology, and our technology has advanced quite greatly in this area. So we can get large amounts of raw data of just sort of the letters that make up this genetic code.
Yeah.
But then the next step, we talk about bioinformatics and the genomic stuff, is understanding, well, what does that mean? So we have this, it shows us the variation that, at this, organism A, individual A has, ATGC here, and the next one has ATGG. Like how does that then translate to what you said, a phenotypic difference? And that's, I think, where it's like it gets a little, we have a lot to learn in terms of, okay, so the letters are different. That means the gene is slightly different. But there's also things like gene location, with, what genes are next to each other. And that can influence how they're expressed, and then what the traits of an organism is. So really, like, genomics, bioinformatics, you guys are digging into this, not just physical variation in the actual base pairs of the DNA, but there's all of this other stuff.
Yep.
And I think that's really fascinating, and maybe people don't grasp like just how much there is to understand there. So how do you go about, like, going from this raw data output, ATGC,
Yep.
to then sort of building this map or this picture of a genome and how it interacts? And even to some level, I'm assuming, once you understand that, you can understand, maybe make some predictions about how it might evolve.
Yep.
In terms of in the face of different pressures.
Yep. No, that's a really good question. So, I mean, to start with, what we're trying to do is find the differences among the A's, T's, G's, and C's that are encoded in your DNA. And we do that nowadays with something called high throughput sequencing. And so sequencing is literally taking the DNA, chopping it up into little pieces, and we have these machines called a sequencer that there's a few different flavors, but perhaps the main workhorse are the Illumina sequencers. Illumina is a brand that is a bit like a big fluorescent microscope that reads these individual bases, which are labeled with a fluorescent probe. And it does this at scale. It can literally do millions to billions of short little sequences at one time. And that's a real contrast, even 10 years or 20 years ago, where this was a much, much slower, much less throughput process. And so what you're left with after your high throughput sequencing run is finished is millions or perhaps billions of little jigsaw puzzle pieces that you need to try and make some sense of. And these jigsaw puzzle pieces are often in maybe 100 letters long, so 100 A's, T's, G's, and C's, 100 or maybe 200. So they're quite short. And a genome is much, much bigger than this. A genome would be in the size, so a typical worm genome might be 100 million or a few 100 million letters. A human genome is a bit over 3 billion letters. Okay, and so this is a really big space. This is not something that you can manually, by eye, scroll through very easily. So what we do is we use computational approaches, bioinformatics, to basically train computers to find where these short reads go. So one of the common, if you've got a reference genome, so this is a genome that someone else has sequenced and assembled, what you might do is try and map your short reads against this genome and look for where in the genome your reads from your sample differ from the reference genome. And then we call these polymorphisms or variants. So there's the variant term again. And these variants might be a single nucleotide variant, so just one A, T, G, and C in a given period, given space, be different. And what we do is we train computers to find those. But it can be much more complex. It could be not a single nucleotide variant, but it might be an indel, so an insertion or a deletion. These also could be a single nucleotide, but they can also be much bigger. And there can also be other types of variations, such as copy number differences. And so often what we're doing is we're comparing our sample to this reference genome and asking what's different about it. Mm-hmm. If we're lucky, we know where all the genes are. So there's some bioinformatics done to try and find the genes. Often a genome, it's worth thinking about a genome, and people don't appreciate this, is a hypothesis about what it actually looks like in an organism. In reality, it's different. It's it's almost always wrong to some degree. It can be very fragmented. So in our cells, we have our DNA that's wound up in chromatin, and that chromatin is arranged into chromosomes. These are really condensed structures that takes really long pieces of DNA that are much, much bigger than your cell in terms of if you unraveled them all. And condensed it to a point at which it could be packaged within this tiny cell. And what the aim of one aspect of genomics, which is assembly, making it a genome, is to try and recreate that structure bioinformatically. But for the most part, that's very, very difficult to do. It's very easy to sequence a genome. It's very difficult to assemble a genome into a representative chromosomal state. It's getting easier to do that. The technologies are really new technologies in the last 5 years or so have really changed the rate at which we can generate those better assemblies. But again, they're still a hypothesis. They're still the best we can do, and over time we'll get better at doing those. Mm-hmm. So we're often
Oh, sorry. I was just going to say this is kind of maybe a shout back to the benefit of preprint, because a lot of this work, as you're saying, everyone has to kind of compare their work, right? Like to get okay, we've all sequenced this worm, this species. What did you guys find? How does that look in comparison to ours? Yep. Not just to sort of find the differences, but also to begin to piece together this larger, which piece goes where?
Yep.
Where does it start? Where does it end? That kind of thing. So you need all of these people kind of working together on that. Just to jump in.
No, there. Definitely. And I guess part of my passion for open research really comes down to the use of genomes for when I started doing genomics. And so this was back in Australia. I was working on a filarial parasite, so a parasite of humans called Onchocerca volvulus, and we were trying to identify genes that might be associated with a suboptimal drug response. So it looked like in some parts of Africa it wasn't responding to the drugs that are used to treat it so well, and so we were trying to do a genomics approach to find this. And our starting material, not the parasites that we were using, but our genomic reference came from the Wellcome Sanger Institute that was made available to the public before publication. And so my research immediately benefited from the work that they had done and released without really restriction. And we wouldn't have made nearly as much progress without that simple release of data before it was published. It's not peer reviewed, I had to use it responsibly and understand that there was caveats in the use of it. It was still in lots of pieces, but over time it actually improved, and I redid my analyses um, and made it better. But fundamentally, it was the open access sharing of genomic data that allowed me to do my research well before I had any affiliation with Sanger. And in
Mm-hmm.
fact,
Mm-hmm.
all of our data that we generate now is released publicly. All of our sequencing data that we generate on our high throughput sequencing machines is released to the public, usually within a few months of it being generated. That doesn't mean that everyone would be able to use it and in the same way that we would use it, given that we generated the data. But with one of the species that I've worked on perhaps the most in recent years, a nematode called Homunculus contortus, which is the barber's pole worm that infects sheep and other ruminants. We submitted our genome assembly and all of the genome annotations to be made publicly available before that paper was published, because really the benefit is to the community is not the publication at all. I mean, the publication is my interpretation of what the data says and why I think it is interesting, and we've identified some novel aspects of the parasite biology. But really, I think my biggest contribution, in fact, is simply just releasing all the data so that people can use it.
Mm-hmm.
And already, we published that paper at the end of 2020, and already it's been cited numbers of lots of times because people are using those data sets before the publication was available.
Mm-hmm. Yeah. And this is where I think, preprint is especially good,
Yep.
because genomics is such a collaborative thing. And again, to put it in the context of the pandemic, coronavirus, there's like a real urgency to understand, what do changes in the coronavirus genome mean?
Yep.
And what is that going to mean for its ability to infect, transmit, that kind of thing, be more deadly, that all that
Yep.
stuff. So it's really it kind of feels like it's one of those fields that sort of really needs to be super open, like for the sake of the research itself.
I completely agree. I mean, I think had all of the results been hidden behind journals, then the response would have been a lot slower in terms of the genomic epidemiological response.
Mm-hmm. But I think even too, like we're seeing the value of this technology. It's finally kind of getting its sort of big moment on the stage. I mean, not to like cheer the pandemic or anything like that, but this genomics has been building. This field has been building for like the last 10, 20 years.
Yep.
And it's really getting to a point now, I feel, where it's like, if we can very, grasp, what genomic changes mean, like when a gene changes, what is that going to mean for the as you said, the phenotype of the organism? How can we, in some, people always say, oh, we're going to play God and whatever. But if you can understand how this process works, what environmental factors will lead to, a response, the genome evolving in one way or another.
Yep.
This is really a big frontier, I think, for a lot of the issues that we face in terms of, drug resistance, pandemics, even conservation and climate change. Some people would disagree with this, but I'm kind of the mindset that like the cat's out of the bag. We
Yep.
change our environment all the time. So now we're going to have to look at making, maybe directing evolution a little bit in terms of allowing us to deal with the changes that we've made.
Yep.
So to me, genomics is really kind of the next big sort of, scientific frontier. And if you look at the other fields, physics, people say, oh, physics is kind of stagnated. We've been working on Einstein stuff forever and nothing really new has come out of it. This area of biology and understanding the genome and how to use it and what does it mean for the things that we can do or the diseases that we can cure, or that kind of thing. To me, this is like really one of the fresh, new, exciting, very active fields. And the pandemic is kind of just highlighting that.
Yeah, I completely I completely agree with you. I think it's really exciting that, and I'm excited to see what the follow-on effects will be of the fact that lay people are talking about variants and genomes,
Mm-hmm.
and I think that not everyone gets it, which is perfectly fine. But the fact that is part of the discussion, I think it's been really great, and I think this and Ebola outbreaks, there's this growing trend towards fast genomic epidemiology that is really being useful, and I think it's been it's genomics has been around for a while, and we've been using it for a while in different things, human medicine, all sorts of different uh, uh, all sorts of different uses for genomics. But perhaps this is one event that has captured people and has perhaps got people to understand a little bit more about it. It's being used to track malaria really quite effectively. It's been used to track other disease outbreaks like cholera. It's being used to track antimicrobial resistance in hospital settings. It is used very, very effectively on some of our most important pathogens. But this this COVID sequencing is really shown that how well it can be used at scale,
Mm-hmm.
and
And maybe
all
we
of it
can.
is because of open data sharing, really.
Right. Yeah, exactly. Internationally
Mm-hmm.
and all of that stuff. Yeah. Maybe we can try and unpack that a little bit. This sort of idea of, genomic epidemiology. Epidemiology broadly is trying to understand, how a disease is moving through a population.
Yep.
Why is it making people sick? And then, using that information to sort of, stop the outbreak, right?
Yep.
So what specifically, like, from my understanding of it, genomics just give us, rather than having to look at the outward symptoms of a person, okay, this person's coughing, they were in a room with this person, so we kind of assume that they got it from them,
Yep.
that this contact tracing, another idea that people are probably now familiar with when maybe they hadn't been before. But if we can take samples from each patient that has gotten sick with SARS or malaria or something like that, we can then look at the genomes of those two, the virus or the parasite infecting these two different individuals, and start to maybe say, answer some of those questions. Get a direction maybe of like it transmitted from one person to the next, or it's changing in this way. And so how do you kind of, what would be the clues, I guess, that you would look for to sort of map those questions?
Sure. No, it's a really good question. So, really, what we're looking for are these variants, and organisms over time will accumulate variants. In us humans, we inherited some DNA from our mothers and some DNA from our fathers, and that's why we look a little bit like them, but also we look a little bit different. Part of those differences is we would have accumulated de novo variants, new variants that are not present in either our mother or father, and that they'll also be different from our siblings as well. And so what the genomic epidemiology is trying to do is look at what variants are shared between different samples, if you like. And we know that some parts of the genome are more likely to change than others. In effect, mutation is a random process to a degree, but evolution, the selection of those variants, can change their frequency. And so what we try and do is understand what's the likelihood of seeing those two variants between two different individuals. Often there might be more variants, and individuals that are very closely related. For example, if you have a transmission event from one person to the next of the virus, you would expect that those two groups of viruses that you sample are going to be very closely related. So over time, we know that mutations accumulate at a particular pace, um, a mutation rate, if you like, and that tends to be fairly consistent. Can change, but tends to be fairly consistent. So what we can use is that mutation rate over time, as well as sort of the spatial sampling where samples are collected from, and try and understand what are the relationships, what are the genetic relationships. So it's a little bit like making a family tree, if you like,
Right.
of viruses. And now there's over 1 million SARS-CoV-2 viral sequences available to make in this family tree.
Right. So you have, just to kind of like, I'm just trying to think of all, the stuff you said, and you said virus, DNA, whatever organism, it's going to mutate at a certain rate. So just even if just over time you have, a virus starting at this point, you can kind of estimate, at a week later or a month later, we expect X number of differences between this genome and this, because it's replicating all the time, right?
Yeah.
So in that sense, you could say, okay, we sampled the virus from this person in this city at this date, and then another person, on another side of the city or in another city at another date, and then use that kind of time frame of how many mutations you would expect to see to sort of say, oh, there's a linear pattern here with geography and the mutation accumulation and stuff that says, well, it looks like, person A from this city probably brought the virus to this city, and it spread from there. And then beyond that, you can then look at, oh, well, there is some weird difference. There's a difference that we wouldn't expect to see just with this sort of stable rate of mutation. And that's maybe where you could be like, it's evolution has selected for this particular change, and it's kind of taking off, and this is where we get the variants, that kind of thing. Is that
that's effectively how it works. I mean, there was an estimation of, I think, something like 1,300 introductions into the UK in the early stages of the pandemic, in between when there was talk of shutting down the country. Yeah. And these would have formed clusters, and some of those clusters would have made sense in terms of pinpointing other little hotspots that had been appearing in Europe and other parts of the world. And
so you could say this cluster that's arrived in the UK looks identical or next to identical to, say, a cluster from Italy.
Yep.
And then you're like, okay, likely. And that's where then you look at the travel records and everything.
Yeah, that's exactly right. So it's it's one piece of information that does need to be pieced together with other parts of information. But that's how it has been used. It does get much more difficult when, in really high levels of transmission and the pandemic, these sorts of things start to not work as well. But they were very effective, for example, of identifying the British variant, B117, I think it's called, and looking at the rates at which that started to take off. And it was through the genomics that identified that particular variant and the increase at which those variants were being transmitted.
Mm-hmm.
Now it's
so
hard
you
to
get this like, sorry, continue.
I was just going to say, hard to know how a variant will behave, but essentially what will happen is that a variant will increase in frequency if there's some degree of fitness increase, and that might be transmissibility or something that allows it to spread quicker, and that will change the rate.
Mm-hmm. Yeah, so you have this clue that, okay, this new cluster of sequences of virus that we're sampling from a bunch of different people in an area, it looks different. So just on the surface, we can say, okay, there's some sort of change, and we can try and map that to its next closest relative to get a picture of where it's being spread to. But then you can also look at where in the genome it's different, and how that might affect its ability to infect. So, for example, again, everyone's heard about the spike protein by now, right? This protein that the virus uses to latch onto cells and get in. So, if you started to see mutations in that area, like this one cluster all has this specific change in the spike protein, then that gives you a clue. Oh, well, this might have something to do with its ability to transmit or its ability to survive in the cells, that kind of thing. So this is another angle that you then, it's not just mapping the spread and sort of tracing it and finding it and chasing it around the world, but then you get, clues as to function, like what it's going to do.
Yeah, exactly. And I guess it allows you to build new hypotheses about what it might be doing. It's very difficult to say anything conclusively until you test it specifically at a lab with a specific experiment that's designed to test that specific variant under certain conditions.
Right.
But, obviously spike protein variants are a major concern, particularly around the effectiveness of vaccines and such.
Right. And so then with your work specifically, you're looking at variations in genes in the worms that respond to the drugs that we use to kill these worms or keep them into control. And in that sense, you can look for, okay, how is because drug resistance is this constant battle between we come up with a drug that kills, a worm, in sheep, and then X number of those worms survive because they have a slight, genetic mutation that allows them to survive, and then the next generation is filled with that type of worm that's slightly changed, and over time it becomes this arms race, right? That's the general principle. So you are looking at sequences of worms from all over and trying to find these sort of little differences that are allowing the worms to better survive the drug. Is that correct?
Yeah, that's a pretty good overview of what we do. So, I'm interested in drug resistance among other things in terms of variation, but drug resistance is a really important aspect of control of these parasites. So it's a much bigger problem in the veterinary fields, particularly in our livestock industries, but it's also a growing concern in the control of human infective parasites, in that it seems like constant use of drugs is forcing the parasites to evolve to be less responsive to those drug treatments. Mm-hmm. And so we take a couple of approaches. One is to sample widely, and we've done some global studies of parasitic variation to try and understand and characterize all of this variation within a species, and that tells us that they're extremely variable. And one of the problems with that is that we're trying to find a needle in a haystack, if you like, in that we're trying to find, probably will be a single base change, so a single ATGC change in about 300 million A's, T's, G's, and C's, right? And because they're really genetically variable throughout the world, and even on a single farm, if you like, if we were talking about our sheep parasites, they're so genetically variable that understanding what that single causative variation, that drug-resistant variation, understanding what that is and what that looks like in a vast sea of all this other variation is really difficult. So we use an experimental system in sheeps where we can do what's called a genetic cross. We take resistant parasites and we take susceptible parasites and we surgically implant them into the abomasum of the sheep where the parasites live. That forces them to mate. And then the progeny that we get, the little baby worms that come out in the feces, are a mixture of those two parental strains. And what that does is it kind of controls for all of that variation that really confounds these studies of drug resistance, and then becomes much less of a problem. And then we do some drug selection experiments where we treat with and without drug, or we treat before and after treatment, and then we use genomics to compare that before and after treatment or between different drugs. And It's been a really powerful way of mapping, finding out where in the genome these genes that cause drug resistance lie. And yeah, it's worked really quite effectively.
Mm-hmm. Mm-hmm. Yeah, that's an interesting approach, and I wasn't involved in that work when I was working with John Gilliard, my PhD supervisor. I was kind of off on my own doing another project, but it always it sounds kind of complicated, to sort of this taking susceptible and resistant parasites and putting them together, having them mate. But I guess the simplest way to think about it is like, and you correct me if I'm wrong, is like they you would assume there's high levels of variation, as you said, between these worms, right? And we don't, some of that leads to meaningful changes, some of it doesn't. It's just the way that they are, right? So by crossing them, by having them breed, the susceptible, the ones that can be killed by drugs and the ones that are resistant to drugs, you're kind of like canceling out all of that variation, so that the things that's left would be what is important to being able to resist the drug. And then when you give them drugs, and the ones that die obviously were susceptible, the ones that survive are resistant, you're left then with this sort of, you can kind of look at, well, the background, all that background noise kind of gets canceled out. And so then the thing that you want to see, the trait that you want to see, you can really sort of hone in on the differences.
That's exactly right. I mean, it's taking two bags of different color marbles, throwing them all together, mixing them up, and and what happens through sexual reproduction, which is why we have sex, really, is to generate new combinations of genetic variation in our progeny. And by mating the different strains, we're basically mixing up all of their genetic variation, so that rather than being separated by strains, now they're all together. And yeah, when we treat them, only the resistance one survives. And what's important is that all of those resistance worms will share a very small part of their genome. They'll share lots of their genome, but they'll all be very consistently sharing a very small part of their genome that contains the resistant variants, if you like. And
Yeah,
so what we
the
can,
the gene that allows them to survive drug, it'll be very like, it'll kind of stick out in this sea of variation as a
exactly,
as a region that's like almost identical in all of these guys.
that's exactly right. It'll be almost identical, and it will look very much like the resistant parent at that particular position. Whereas the rest
Right.
of the genome won't. It will just look like a mix of the two. So we can use these bioinformatic approaches to compare pre and post treatment, or the susceptible versus the genetic cross treated individuals, and it's been very very effective. I think one of the really interesting things is that we've been able to identify a number of known drug resistance genes, so it kind of validates our approach. But we've also been able to exclude a number of drug resistance genes in the literature because they just show no evidence of selection in these strains. But we've also been able to identify some novel things, some that just are completely off everyone's radar, which is quite exciting too.
Mm-hmm. Yeah, and I mean, like, this then gives you, so that you can look at the changes. You could look at the gene that's providing this resistance. You can then, you know, understand how it does the resistance, maybe how it's conferring that ability to survive the drug. I know in some situations that there's like it codes for like a pump that maybe gets rid of the drug, or it provides a blocker on the outside of the cell so that the drug can't bind and get in. There's all these different things, so you get that information. You find things that you never expected to find. And then you can also then, in your global sampling, now you have a target to look for, right? To say, hey, resistance is growing in this area versus this area. So maybe we need to like either find new drugs. So it gives you that information too. If you know how it's beating your drug, you can design a drug to get around it in theory. But you also then understand, well, we need to not use as much drug in this area because otherwise we're just going to keep breeding these sort of like super worms, right?
Yeah, most definitely. I think one of the sort of the short term promises, I think, it hasn't eventuated. I think we're pretty close. Is having some diagnostic tools that will allow us to predict what level of resistance might be on a farm or in a population, and then use that as a management tool to say what should we be treating with, and whether we should be treating with the same drug or whether we should be switching to a different drug. One of the issues is that there's a very few number of drugs available, and so both in the human and veterinary settings, managing drug susceptibility, if you like, is really, really key, because there's not too many options. If all our parasites become resistant to the very few drugs that we have, it's going to be very difficult. And in some places in the world, farming, for example, has become uneconomical because of the high levels of drug and subsequent drug resistance. It's just very difficult to control these parasites. That obviously has significant animal health and other economic burdens, if you like.
Mm-hmm. Well, and I think that's maybe something that you could fill us in on, because I think people don't realize, we think of parasites. People know viruses, especially now. Bacteria they hear about a lot. Bacterial resistance, which is essentially, people are doing the exact same thing that you're doing with worms in bacteria to get to find out about bacterial resistance. But we don't think about parasites as much unless it's like some clickbait video on YouTube of like a gross parasite or something, or unless you have dogs and you've seen them have worms, or cats or something like that. But I think people don't realize just how, especially in, sort of high income countries, how important these things are to, livestock management. And then in low and middle income countries, it's even, the problem, is even worse with their livestock, because they depend so much on it. They don't have the sort of same systems and wealth that we have in order to sort of deal with these things. So if livestock gets sick, what are you going to do? And then also, in the low and income middle countries in tropical and subtropical areas where these things tend to really thrive, it's a human burden as well. And the parasite thing, again, just, people don't really understand it. It's not so much about them killing you, but it's them making you really sick, or your animals really sick and unproductive.
Yeah, no, definitely. I mean, I think it probably hasn't gotten on a higher priority or radar as something like malaria or other bacterial diseases, because it doesn't kill a lot of people. It's estimated that probably 1.5 billion people worldwide have a parasitic infection of a worm. And these are almost all in low and sometimes middle income countries, some of the poorest regions of the world. And often they're infected with more than one species. And so they have a really high burden in some regions of the world, and they can make people quite sick, including children. I mean, it's some of these parasites, particularly a group called the soil transmitted helminthes, really have a big effect on children's development. It's proposed that might have some impact on cognition and other developmental issues. And so, it's really important. And the reason, I guess, well, one aspect of why they're important is that they are recognized as being something that we need to control, and they are actually targeted by some of the world's largest drug delivery campaigns, mass drug administration. A lot of these drugs are donated for free and delivered into these really poor endemic communities to control these worms. But in some parts of the world, these drugs are not necessarily working as well as they should. But in terms of farming, effectively all farmers, as part of their management program, treat their livestock with drugs to control gastrointestinal parasites. And so it's just rife. And I think one of the real challenges is that there's not really a standard way of managing livestock in terms of parasite control. It varies from region to region. It varies from what your father or grandfather did. There's so many different ways of controlling them, and there's probably not always a great understanding of how to use drugs properly and underdosing or overdosing or not treating enough. There's all these different factors that can lead to the evolution of resistance.
Mm-hmm. Mm-hmm. Yeah, and I mean, just we can kind of start to wrap it up here. But the you mentioned how parasites are so variable, and it's one of those things that, like I said, people don't really think about much until it's like a gross video of a worm or
Yep.
something, or their pets and things like this. But they're really beyond the practical matters of the burden in low and income middle countries. And you said it doesn't matter where you are, every farm deals with these worms.
Yep.
Like it's all over the world, and they do have a cost in terms of production and health of the animals and stuff like that. But these are such fascinating creatures that we can actually, I think, also using some of these genomic techniques. You talk about the novel findings, you find some things, or you find some genes that these worms share that you're like, oh, that's weird. Why do they all have that? Like, it must be important, right? But if you think about where these things live, how some of them live in the bloodstream, in your body, that kind of thing, and the size of them, they're not, they can be big, like they're actual,
Yeah, they're not small. Some of
physical
them are not small.
animals, not
Yeah.
like, yeah, not like microbial things,
Mm.
right? So they have to develop these really sort of intimate connections with the host and the host immune system
Yep.
as to how they get around that. And actually, we can probably learn something about, how our own immune system works by looking at the ways in which worms sort of evade the immune system. And so there's all of these cool little things like traits that they have that using genomics and stuff, you can start to understand, oh, well, hey, like, look at that. This allows this worm to, survive better here. And then you can start to be like, well, if we can, CRISPR that, or, something like this, like it really, it shows you a lot about yourself by understanding how this thing infects you.
Yeah, most definitely. I mean, there's this active research in a number of different areas and ways of exploiting the way worms hide from the immune system. So some of these worms live in our bodies for years. Onchocerca volvulus, the filarial worm I talked about, that causes the disease river blindness. Its adult worms can live up to about 15 years in these little golf ball sized nodules, and they do that really effectively, hiding, suppressing the local immune system to trick it into not knowing that it's there. And so really understanding how these parasites are so good at at hiding within the body could lead to so many novel products that could help us in a range of different other diseases.
Mm-hmm. Mm-hmm.
Lots of inflammation or asthma or gut diseases. There's a number of different ways people are trying to explore using Helminth-derived products for that.
Yeah. And I mean, people may have heard of like this, the idea of like taking a worm to help your gastrointestinal, disorder, IBD or something
Yep.
like this, autoimmune disorders, basically. If
Yep.
you have a thing that suppresses the immune system, like very locally,
Yep.
so it's not going to affect your whole body. It's going to be sort of in this you could see how that would be useful. But then it's not great to just infect someone with a worm, right? Like, because there's other complications that could come out of it. But if you can isolate that using genomics and say, hey, this gene makes this product which does that action, then you can take that gene, grow it in the lab, and sort of turn it into a drug, really.
Yeah, I mean, I think there's lots of different sort of approaches that you could use to sort of get to that. But that's fundamentally the idea, is it better understanding of these gene products and what they do, how they interact with the host. All this sort of work could ultimately turn from having to give someone a worm, which not everyone would be so excited about, perhaps swallowing worms, but some again, it depends whether you've been living with a debilitating illness for a long time. You would probably be very open to that. But even better yet, if you can identify what specific molecule is important for the worm's survival, then that might be a great drug candidate for doing that without the worm.
Mm-hmm. Yeah, yeah. And just in terms of like basic research that you don't know, you never know where it's going to go. I know, people might not know this, but there's, you mentioned nematodes as a type of worm that you work with, and they can be parasitic, but they can also be not parasitic,
Yep.
and they just sort of live in the soil and stuff. And the nematode C. elegans is like, it's been such a resource in terms of just understanding development.
Yep.
So you look at this tiny little worm, and because it's been so well sequenced and we have a really good map of its genome and stuff, people can do experiments that relate to how all organisms develop from, sort of the single cell stage all the way up. How do tissues develop? How
Yep.
does, nervous tissue differentiate from muscle tissue and stuff? So there's this, again, genomics playing such a huge role in where we're going with medicine and understanding our bodies and stuff. But then also these little worms, that you'd never see, you'd never heard of.
Yeah, I
It's
mean,
fascinating.
it's been such an important organism for even human studies. There's a huge number of genes that are shared, at least some degree of function. So there's been really fundamental insights into human biology through studying this tiny little worm that is literally everywhere on Earth. If you basically stripped away all of the land and left all the nematodes behind, you would still kind of see the Earth effectively, the hills
Yeah,
and the mountains.
there'd be a blanket of nematodes.
There'd be a blanket of
like
nematodes.
wherever
It's
there
just
was
everywhere.
soil, wherever there was water. Yeah, yeah. It was one of those things when I got into parasitology. It was kind of by accident, right? Like I was going to be a school teacher. I was studying. I was always into biology. I was going to be a school teacher, and then kind of realized I didn't really want to be dealing with children all day in that setting. So I was like, okay, so I'm going to just chase the biology instead of the school teaching thing. And I just had a class where the professor was really into parasites. That's what he studied. I think it was even just like, it wasn't even a parasite specific class. It was just invertebrate zoology.
Yeah.
And I was looking at these things and the way he was talking about it, I was like, that is just, it's incredible. I mean, they're everywhere. Parasites, it's probably one of the most successful forms of like lifestyles
Yeah.
on the planet. Like everything has a parasite, no matter what. There's something that parasitizes it. And then, yeah, like these, such intimate relationships with the coevolution. Then you get, the behavior manipulation aspect that can happen and stuff like this. And so it's like, that right there is fascinating. And to now then have the tools to sort of look at how it does that on the sort of molecular level, how is it manipulating behavior of a species? How is it evading the immune system? And looking at these genes and how they change and what does that mean for the cell structure and stuff. Like, it is just, I think people really are starting to realize the if we can grasp the genome and understand what that means and then apply that knowledge, the things that we could be able to do is, people, again, physics and stuff always gets all the play in the headlines, black holes and all this stuff. Oh, we're going to go to Mars and we're going to live on Mars and stuff. Yeah, well, without genomic information, how are you going to develop plants that can live on Mars or terraform the planets and like all? I mean, we're getting way out there, but that's the kind of like, that's the stuff that it's obviously way in the future, and who knows if it's good or bad. We can argue the ethics of it, but this really is like a really amazing frontier of knowledge that we're on.
It's a really exciting time to be a part of it. It's certainly becoming much cheaper to do these sorts of experiments, even though they're still very expensive. But it is certainly getting cheaper, and I mean, it's driven. I mean, we benefit in the parasitology field by having other fields, human genomics, really pushing the technology and the costs down, and it's this trickle-on effect to not only parasitology but all other non-model organism research that we benefit from these technologies and analytical tools and all this sorts of stuff that you just couldn't imagine even doing 5-10 years ago. We could just do so much at scale now, and there's just really, fundamental questions that we can tackle now that would have been much harder to do before.
And I mean, it's only going to increase, right? Like you have the sequencing machine itself and how that works. You gave us a little description of that. But then there's, the bioinformatics side, which is computational power, algorithms, machine learning, all of that kind of stuff that plays into it. So it's also a really neat, merging point for all of these different fields. And so for people that, young students that might be interested in this kind of stuff, it's like, did you ever think you could take your programming computer background and end up working in biology on worms or something?
Yeah, 100%. I mean, I think now it's getting to the point where it's just so easy to generate the data. It's still a really tough ask to analyze it effectively, and new ways of doing it all the time. I think it's exciting from a number of fronts, from both methods development to bioinformatics or computational biology or algorithm development. There's just so much that can be done, and I in fact trained as a molecular biologist, and I didn't do any parasitology. I only started parasitology in my postdoc. So I trained as a human molecular geneticist. And it was just through, I don't know, good or maybe bad timing, but good timing for me that a position opened up, and that was working on parasites. And I thought, I'll just go learn a bit of genomics and I'll come back and work on humans again. But I've been hooked ever since.
Yeah.
And there's just so much scope, so much to be done in this field. And it's a really good time to do it.
Yeah, amazing. Well, I'll let you go. I appreciate you taking the time and breaking down some of these things for me, and giving your thoughts on communication and the preprint stuff. Some things I didn't think about, especially as it relates to, equitable access in terms of low and middle income countries and stuff like that. Really interesting points. So thank you so much for being here. I really, really appreciate you taking the time.
No, my pleasure. It was great fun.
Okay. Well, and we can do it again. I would love to continue. There's still so much more to talk about, so we can definitely do it again. And then just quickly, you can, because you're pretty active on Twitter, I find, and it's always nice to see you commenting on the different things you see, the preprints, you give your take on some of that stuff. So it's, for people that are in the field or even just kind of want to see some conversations that are going on about it, I would encourage you to follow Steve on Twitter. I'll let you give the Twitter handle.
Yeah, so it's Stephen __ Doyle, the subtle double underscore. But do follow.
Right on. That's great. So Steve, thank you again very much, and enjoy the rest of your afternoon.
Thanks, you too. Cheers.
And there we go. Again, many thanks to Steve for joining the show. It was a really great conversation. I really, really enjoyed it, and hope to do it again. As you just heard, you can follow Steve on Twitter at Steven__Doyle. Follow us on Twitter at 2bradforyou. Also on Instagram at 2bradforyou. Send us an email, 2bradforyou@gmail.com, or leave us a voice message, speakpipe.com/2bradforyou. Rate us, subscribe, all of those good things. We so much appreciate it. I think that was terrible grammar there, but we do appreciate it, and we will see you next time, everybody. Stay safe. Love you lots. Bye for now.